Welcome back, welcome back. Let's hit present. And welcome to the third module. Hopefully, if you're watching this, that means that at this point you already took the second exam, and I hope that went well for you. And now it's time to get into some business on the third module, which we will dub Sensation and Perception. In this unit, we start with chapter 9, which is the auditory system and hearing. And so we will get into the pathway that sound waves take. We'll talk a little bit about sensation and perception, how our nervous system takes something physical from the environment and turns it into a signal the brain can understand. And we will finish off the second half of this lecture after talking about language, because hearing and language are very similar, using similar parts. Language actually brings together the visual and auditory system, and that's why if you were to for the language portion of the chapter. So this is usually in 3 parts because I go over the auditory system twice. I won't need to do that if it's recorded, obviously. So this will take us through the end of next week, and there will be an assignment coming up for that. But this week, since you guys are taking exam 2 and then watching this, I'm not going to assign anything. Come on, there we go. Did I miss one? No, I did not. Just so when a system in terms of sense outside of the brain and spinal cord into the brain to be processed, we talk about this specific system. When we went over chapter 2 and how the nervous system communicates, we talked about receptors being the little locks and the neurotransmitters being the key on the dendrites. But in the sensory system, we also use the word receptor. And a receptor, in this case, is a cell, not a part of a neuron. It's an actual cell itself that basically takes the stimulus energy from the outside world and converts it into a signal the brain can understand. And as we talked about in chapter 2, the signal that the brain understands is action potentials and chemicals. So in the 5 sensory systems, we have 5 specific receptor cells. As you can see on the slide, they respond to a particular form of energy that we call the adequate stimulus. So in the eyes, which we'll talk about in chapter 10, the adequate stimulus is light waves. For the touch system, the adequate stimulus would be pain, temperature, and pressure vibration type of touch. For taste and smell, they are chemicals that get converted. And of course, in the auditory system, which is what we're talking about right now, they are sound waves that come into the ear and kind of vibrate off of the bones and parts of your ear. And then inside the inner ear, as we'll talk about, they get converted into a physical stimulus. From a physical stimulus to a neural stimulus. And what that means is the eyes will only convert light waves, the ears will only convert sound waves, etc., etc. You can't shine a flashlight into my ear and the parts of the inner ear are going to go, oh, that's light. Let's convert that for the eyes. That doesn't work. You also can't just cut open the brain and shine a flashlight into the occipital lobe in the back and have the occipital lobe neurons go, oh, that's light, because they can't actually read the physical light. It has to be transformed, or what we call transduced, into a neural signal. The brain can only understand the patterns of firing, of action potentials, and chemicals. That's the language the brain can understand. Um, so, as we also went over in chapter 2, we know that the size of the action potential is always the same, whether it barely reaches the threshold um, or whether it goes way over. Doesn't matter. Remember, the all or none response means the action potential is always the same size in firing. So, what makes the information meaningful is the pattern and intensity of how the firing goes. More action potentials is going to be a stronger signal. The wavelength or distance from peak to peak of these waves is going to be make the difference in how high or low the pitch is in the auditory system. In the visual system, it tells you what color you're seeing. So we'll go over that. You'll see through chapters 9 and 10 that there are a lot of similarities in the parts of the brain and the wavelengths and how the visual and auditory system interprets these waves, the type of stimulus, they're both waves, and things like that. So the intensity and the pattern of the information is what the brain is interpreting and basically is turning into the signal that we perceive. So it's good to talk about right now for me the difference between sensation and perception. Sensation is basically the individual little pieces of information that are coming through the waves that the brain is interpreting. The ear gets the information and sends that information to the brain, and that's how we make sense of our outside world. So all of those little individual pieces of energy that the brain has to code and put together, that's what we call sensation. It's the acquisition of the information. So we call this a bottom-up process because it's almost like your brain is taking the little individual pieces of information, sending them up through these neurons, through the receptors that change them into a brain signal, and then the brain is taking these little pieces and putting them together into the big picture to interpret what it is that we're hearing, seeing, touching, tasting, and smelling. So we call this a bottom-up process with the individual little pieces. So I like to think of it like a puzzle. When you have a puzzle, you take each individual piece and figure out how it fits together into the big picture, right? The big puzzle. So you can think of sensation and this bottom-up process as all these individual puzzle pieces that are traveling up to the brain individually, and as soon as they make it into the brain basically puts the puzzle together into whatever this is. So that's how we interpret what it is that we're hearing or seeing. Perception, on the other hand, is how we interpret that information. So if you think of sensation as the little individual puzzle pieces, think of perception as putting the puzzle together and then comparing it. This is a top-down process. So we put the puzzle together, we understand what it is that we're hearing or seeing or all of the above, and we compare it to our memories, we compare it to our expectations, we compare it to things that our brain has already interpreted. And this gives us a very individual, personalized perception. So, if you've ever heard the term perception is reality, that's because we think of it as tailored to each individual person and their brain. Um, so, in that way, the sensation, let me give you an example. If we see a dog walking down the street, I usually use at the park. We're not really supposed to be at the park. So two people could be standing there, right? Seeing a dog. This thing that they're seeing, they see it as the same color. They see it as the same shape. They see the four legs, the tail, the ears, the fur. So what they see is the same images. What they hear when this thing barks is the same sound. They hear it at the same frequency. If they're close enough, hopefully not too close, social distancing, they can maybe even smell the dog, right? But person A, which might be me, those pieces of information and sensation go up to my brain. Once they hit my brain and the big picture comes together, oh, it looks like a dog, it sounds like a dog, it smells like a dog. That must mean it's a dog. And that might be the same thing that's going through person B's mind. Now, what we do with that information or how we perceive that information may be specific to each of us. For me specifically, I grew up, when I was born, we had a 6-year-old dog who was already pretty old by the time I could, move around and things like that. I've always loved dogs. I'm a dog person. I've got, well, I had two. One of them is with my brother now. Um, so I've got one main dog, and I am a huge dog person. So I see, hear, smell this dog, and my first instinct, even though social distancing, is I want to really play with this dog, but I can't. But if I could, I would be going over there asking if I could play with this dog. Now, person B could be any one of you guys from the class, could be anyone. Person B sees the dog, hears the dog, smells the dog, and their individual perception might be based on their past that they either have never owned a dog, they're a cat person, they don't care about animals, um, so they may be indifferent. They go, oh, it's a dog, whatever. Keep walking, ignore it, don't even register, right? Or maybe it's somebody that did have a dog when they were little and maybe they got bit, had a bad experience with a dog, um, something like that, had to put a dog down, and so the memory is too painful, right? So their expectations, their memories, and things like that, they see the dog, hear the dog, smell the dog, maybe, and their reaction is either dog, whatever, couldn't care less, or oh my gosh, that's a dog. I'm gonna go the other way. I'm not a dog person. Um, so that's why everybody's perception of things may be a little bit different. And as we'll talk about through these next few chapters, um, due to a person's individual threshold levels, some people are more sensitive to certain things than others. It makes your perception um, potentially quite different from everybody else's, right? So perception is reality is just a way of saying that what is personally useful or personal to you is how you see the world once you put the big picture together. How you interpret that world is specific on your experiences, your expectations, your memories. So moving on, if my computer will let me, we can see that there is a very many similarities between the auditory system and the visual system. For instance, our adequate stimulus in both the auditory system and the visual system are waves in wave form. So when we look at the waves in hearing, it's what we call sound waves, which are sort of waves that are vibrating in a conducting medium as they go through your ear. So they go through air, they hit off your bones, as we'll talk about, and go through the fluid of the inner ear, which we call the cochlea. But they are physical waves of bent and compressed air molecules. In the visual system, we also have waves, which are electromagnetic in the visual spectrum of wavelengths. So in the auditory system and in the visual system, the brain is interpreting the length or distance from the peak of one wave to the peak of the next, which is what we refer to as the frequency or wavelength. As you can see in physical modalities, frequency on the top and wavelength in vision on the top of the vision one. Those are the same thing, frequency or wavelength. Wavelength is technically the distance from peak to peak, and frequency is the measure of how many of those peaks we see in a given time, like seconds, how many peaks per second. But it's basically the same idea, same thing. When we are looking to hear sounds that are louder or quieter, we're not looking at the distance from the peak to peak of the waves. We're looking at the height of a wave. The bigger the height, the more intensity or amplitude of the wave. And what that shows us, how that's interpreted, is a louder or a quieter sound. So as you can see, for psychological modalities, frequency is pitch, That's high or low pitch. And amplitude or intensity is how loud it is. We also have this in the visual system. In the visual system, we also have wavelength or frequency, and we have intensity or amplitude, just as you see, because they're both in waveform. So the difference is in the visual system that the wavelength, shorter or longer, is going to determine what color it is that we perceive. The shorter wavelengths are more of a blue or purple color, whereas the longer the wavelength, the more of a red that we see, and everything in between is the other colors. The intensity in this case is not how loud it is, but it is the intensity of what modality we are able to perceive, which is color in this case, or light itself. So the intensity or amplitude is going to give us a brighter light or a brighter color. For the auditory system, which we're about to go dive headfirst into, the frequency tells us how high or low the pitch is. So longer, farther apart wavelengths are going to be a much lower pitch. And when the wavelengths are very close together, it's a higher frequency, it's going to be a higher pitch, right? Don't ask me to do that because I'm not very good at that. The last thing I wanted to point out is where the stimulus is converted. That's right in the center of this chart, because it's got to be an adequate stimulus and it's a different receptor cell. So for the auditory system, inside the inner ear or the cochlea are these little cells that look like hairs. They're not actually hairs, but under a microscope, they look like little hairs. They are called hair cells, and that is where the signal is turned in from a physical signal of sound waves. And when these hair cells basically bend, what they do is they give off an action potential and send that signal that used to be a physical signal is now transformed or transduced, converted into a brain signal that goes to the brain. So that's done in the cochlea with these hair cells, and they will only convert sound waves. Light waves will not be converted by the hair cells. Vibration of touch, smells won't be converted in the ear, right? Only the adequate stimulus, which is sound waves. In the visual system, in the back of the eye are a few layers of cells called the retina. And in the retina, at the very back of the eye, are these two receptor cells called rods and cones. These are named for what they look like. Rods look like these little rods under a microscope, and cones look very cone-shaped. The rods and cones, as we will get into in the next chapter, are the ones that convert light. When light hits them, they give off a chemical, as you will see in the next chapter. And in response to that chemical, that starts a chain reaction where the signal is now a brain signal. It's now chemical, which is picked up by the next cell, causes that chain reaction that we all talked about of more action potentials and more chemicals. And that makes its way from the back of the eye to the occipital lobe, to the visual cortex of the brain. And what you'll see in these next two chapters is that it's a very similar pathway. In fact, their paths are almost parallel in a couple spots. So let's move on. Hopefully you can see this in the background. I had to kind of move my screen back a little bit because that green on here on my computer is a little hard to see. But at the top left, it says amplitude or intensity, which is measured in millivolts. And again, we're looking at peak to peak. So if you're thinking of a wave, think of a wave that goes whoa, 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 back and forth, up and down, and just the distance from one peak to the next. So as you see in the bottom right, it looks like that with pitch. Seeing the green arrow on the bottom, it says the pitch is from one peak of one wave to the next one. Although the arrow is at the bottom, think of it as the wavelengths are the peak on the top. So the other one is called frequency. Frequency is measured in hertz usually. Sorry, I got that backwards. I just noticed that. Amplitude is the first arrow looking at the height of one wave. How high the wave is tells you how loud the sound is. And so I don't know why it says peak to peak. Amplitude would be the height of one peak. That's where I got mixed up. The frequency is measured in hertz. That's the one going across from peak to peak, and we're basically counting the waves per second. And as you can see, our range actually of what we can hear as human beings is actually quite limited. Our normal hearing range is as low as 15 hertz and as high as 20 hertz hearing range from low to high. 15 hertz to 20,000 hertz. Loudness is the way we hear amplitude or intensity, which is usually measured in decibels. And the doubling of an amplitude is somewhere around 6 decibels. Do you need to know that for the exam? No, but it's nice information. So once again, just to clear up any confusion, the distance from one wave to the next, or how many cycles of waves per second, is the frequency, and that tells us how high or low the sound is that we're going to hear, the pitch. And the height of one wave, which is almost always the same if the level of volume is the same, is the loudness. So wave length or frequency is the pitch, how high or low the sound is. The height of the wave is the amplitude or intensity, and that tells us how loud or how quiet the sound is. So looking at these sort of demonstration of these waves, based on what we just talked about, we can predict by looking at the sound waves whether the pitches will be higher, lower, whether they'll be louder or quieter. So if we look at A and B are the same pitch, right? You can see that the waves are at the same level. If you were to count that as maybe 1 second, we see 3 peaks in A and we see 3 peaks in B about equally apart. So the pitch, or how high or low the sound is, will be exactly the same between A and B. What's the only difference? If you look, the waves are higher, the peaks of the waves are higher in A than B. So A and B would be the same pitch, but A would be a louder volume. For C and D, we see the same thing. We see a lot more waves in that same area than A and B. So if A has 3 peaks, B has 1, 2, 3, 4, 5 peaks. The closer together the wavelengths are, the waves are, the higher the frequency, the higher the sound. So A and C would be the same volume, but C would be a higher pitch than A. A would be a lower pitch. B and D would also be the same volume, which would be lower than A and C, but there are more waves, just like D. So C and D would have the same pitch. C would be a little louder. But in the real world, we don't really just hear pure tones, right? A, B, C, and D would be pure tones in one register. A lot of times we have, in fact, most times we have combinations of sounds, whether it's listening to music with different pitches and frequencies. If you're listening to a guitar, most all of the guitar sounds are chords, which means multiple notes per strum, right? So in the real world, most things that we hear that have different pitches, volumes, tones, all of that would be a lot more complex than A through D. So if you look at E and F, that's more what you'd see in sort of these complex sounds. So it could be an entire band. It could be multiple people talking. It could be somebody talking with machinery and things going on in the background. That's really the way that our brain interprets things. It would be nice if it was all pure tones like the top, but it's a lot more complex than that. So kudos to our brains for being able to do this so effortlessly. Okay, I guess we don't have a picture here. In this one, what we would see is the outline of the ear, and I'm going to hopefully see if the next slide may have it. Okay, so we can't see the whole thing, but if you were to see this figure and this picture, you would see the outer ear, the middle ear, and the inner ear sort of all together. But what it seems is, for these slides, that we do have each individual portion. So if I can find, and I'm sure I can, I'll put on the, with these slides, I'll put a picture of it together. The outer ear is called the pinna. And if you ever wondered why your ear is shaped the way it is on the outside of your skull, on the sides, the lateral sides of your face, that is because it makes a perfect funnel. So when sound waves, which are bent and compressed molecules of sound, as they bend and compress air molecules, um, it creates the wave. And the best way for your ears to pick it up is to funnel it through these silly kind of shaped ears, these pinnas. But the pinna makes a perfect funnel, as I said, to be able to um, bring the sound in, right? It kind of collects the sound in the environment and brings it into your ear so it can be interpreted and sent to the brain in a very useful way. So as you hear, which is that purple little area at the end of the canal called the tympanic membrane, but you guys probably know it as the eardrum. That is also known as that thing if you stick your Q-tip in too far and you hit something physically and it's uncomfortable, you've hit the eardrum. Congratulations. Um, the reason it's called a drum is that when the sound wave hits that drum or hits that membrane, it causes it to vibrate and the sound becomes a little more intense. So you're basically amplifying the sound. And if you didn't know how a drum works, the reason that you can hear sounds and different sounds coming from the different types of drums is because for those drums, the drum stick or mallet or whatever it is hitting the drum head. And what the drum head does is it slightly vibrates, or more than slightly sometimes, vibrates back and forth in its place. And that vibration is what causes us to actually physically hear a sound coming from the drum. Now, once it hits the eardrum, that means that it gets amplified as it goes through the tympanic membrane to those little tiny bones you see in the middle ear. So you can see in this picture that there are three tiny bones that were named for what they look like, as a lot of things were, as I've said in this class. The one in green is the hammer. Attached to the hammer is a little one that looks like an anvil. And finally, beyond the anvil that's connected to the inner ear is what we know as the stirrup, because that's what they look like. Of course, that is their English names. In Latin, when they were first discovered, they were called the incus, the malleus, and the stapes, which means hammer, anvil, and stirrup. Those are in the middle ear. And these bones, these tiny, tiny bones, are actually the three smallest bones in our entire body. So here is a better picture of the middle ear as it connects to the inner ear. What we see here is the tympanic membrane and those three bones, which together are named the ossicles: the hammer, the anvil, and the stirrup, the three tiniest bones in the human body, and they transmit sounds and amplify sounds from the outer ear into the inner ear or cochlea. So what happens is once the sound vibrates off of that eardrum or tympanic membrane, it then amplifies a little bit and bounces off those three bones in the middle ear. It bounces off the hammer and the anvil and the stirrup. And while it's doing that, it is picking up momentum and it is amplifying even more. So now you have a stronger sound wave, because just like the action potential, we need to make sure that it gets to its destination. We need to make sure that it's strong enough to get converted in the uh, inner ear by those hair cells. And we need to make sure that that message, when it gets converted into a neural signal, that it makes it all the way to the brain, right? Um, and then it makes its way from the outer ear through the eardrum, the hammer, the anvil, and the stirrup of the middle ear, and through this little oval window that the stirrup is connected to. As you see at the bottom, it says oval window. At this point, it goes from um, air and bone into the inner ear, or the cochlea, which is Latin for snail shell, because it is wound up and looks like a snail shell, as you can see. In this artist rendering, you can see that they took off a couple of pieces of the cochlea so that you can see inside the cochlea. It's actually filled with, there are 3 canals that are filled with fluid. So your sound waves are first going through the air of the outside world and your outer ear. Then they hit off the bones of your middle ear after hitting the eardrum. And then they go into a fluid-filled inner ear and travel through the fluid, kind of displacing it like a jet ski displaces water, leaving a wake on a lake or the ocean, if you've ever ridden a jet ski. So in the inner ear, through the oval window, there are 3 canals. We see the vestibular canal, we see the cochlear canal, and we see the tympanic canal. In this case, the vestibular canal is actually the fluid in your inner ear that has to do with your balance. So we're not really going to talk about that much, but it's good to know that it's there. So if you ever have gotten a dizzy spell, or if you've ever had an ear infection and it caused you to lose your balance, if you've ever heard that balance is an inner ear issue, that's because it is regulated by this vestibular canal, which gets messed up a little bit when we're dizzy. If you go on a roller coaster and it makes you too dizzy that you can't walk, then it's probably displaced a little bit from that vestibular canal. We're not worrying about balance, but that's why if you ever heard that it's an inner ear problem, that's where that comes from. And if your ear is ever leaking fluid, like if you have an ear infection, that's from the inner ear, right? The tympanic canal is also on the sort of left-hand side. But when we talk about the auditory system, we're only going to concentrate on that one in the middle called the cochlear canal. And that cochlear canal is, as you can see the arrow pointing down to C, is where the organ of Corti is. And so what happens is the signal goes, and I'm gonna trace it all the way back. It goes from the outer ear, then it hits the eardrum, then it vibrates off those ossicles, the hammer, the anvil, the stirrup, goes through the oval window, and now it is inside the cochlear canal of the inner ear, of the cochlea. That's the middle canal. For that, once it goes from air to bone to water, or fluid at least, it picks up even more intensity and it displaces the fluid, kind of like a jet ski does on the lake or on the ocean, and causes little ripples of waves. In the middle of that organ of Corti is what we call the basilar membrane. And the basilar membrane, which means base, is the bottom of that membrane through which the cochlear canal goes through. On the bottom, stuck in the basilar membrane, are these little cells that look like hairs. These are the hair cells. On top of the hair cells is another membrane called the tectorial membrane. Tectum means roof, if you remember from the midbrain in chapter 3. So the tectorial membrane doesn't actually hold the hairs there. The hair cells are embedded in the basilar membrane, but they are not quite touching the tectorial membrane. And what that means is that they're not, they're not attached on both sides. So when the fluid comes and hits the hair cells can bend from the top. So what happens when the fluid is displaced and causes little ripples in the fluid or water, is that those little waves come and hit the hair cells, and they cause the hair cells to bend either a little or a lot. And when the hair cells bend, that's when they give off and basically create action potentials. So the hair cells bend and create action potentials, and those action potentials go down the axons of those hair cells. And the axons, remember, a bundle of axons in the peripheral nervous system outside of the central nervous system is called a nerve. So those hair cells, axons, come together to form what we call the auditory nerve. And now, once the waves hit the hair cells, it now converts that signal, transduces into something the brain can understand. The hair cells bending a little or a lot causes action potentials. The more the hair cells bend, the more action potentials there are that go down through their axons, which become the auditory nerve. And what you will see is that the auditory nerve then carries that neural signal out of the ear and into the brain to find its final destination in the auditory cortex. Remember, that's in the temporal lobe. So, again, we have the tectorial membrane on top and the basilar membrane on the bottom, as you can see from this slide, and the hair cells. Now, to add even more confusion to things, which I wouldn't dare do in Psych 1, we have actually two different types of hair cells. We have outer hair cells and we have inner hair cells. The ones that are bending and converting the signal to a neural signal are the inner hair cells. The inner hair cells, as you can see on this slide, even though you can't see the picture, um, the inner hair cells encode sound into these neural impulses. Well, what are the outer hair cells doing then? The outer hair cells are helping to amplify and sharpen the sound so that we can know exactly what it is. So in order to, right, make sure we get a signal that's interpretable, is that a word? I think it is, that's able to be sent to the brain, we need to amplify it as much as possible. So it's when the sound wave hits the eardrum, it vibrates a little bit and increases its intensity. When it vibrates off the bones, the hammer, anvil, stirrup, or incus malleus stapes, um, when it vibrates off those ossicles, it picks up even more momentum and causes more vibration. And then when it gets into the inner ear through the oval window, it's going through the cochlear canal into the organ of Corti, where those hair cells are, and it is displacing the fluid like, again, a jet ski does to water. So it's increasing its intensity ever more, and then those outer hair cells really help to sharpen and focus and make sure that those waves are nice and intense. When it hits the inner hair cells are the ones that bend and cause an action potential that travels down the auditory nerve, or the axons of many hair cells put together. Now, that's just the pathway to leave the ear. So outer ear to eardrum and ossicles in the middle ear, through the oval window into the inner ear, and down that cochlear canal into the organ of Corti. When it reaches those hair cells, the hair cells bend. The signal is now no longer a sound wave. It is now a neural signal in the form of an action potential that, when it gets to the end of the auditory nerve, will turn into a chemical, of course, right? Opens the gates and allows the chemicals to flow, and now we have a signal that goes to the brain, and the brain can interpret it. So the auditory nerve is also known as the eighth cranial nerve. And for the purposes of learning in the exam, we don't want to get too complex. This is still, although it's a complicated class, it's still a lower division class. So you can see things like in A on the picture, on the illustration, you can see things like the cochlear nucleus and superior olive or superior olivary nucleus. A lot of times we just call that the superior olive. I don't want you guys to get too wrapped up in the details, as these details are already a lot more complex than Psych 1. So if you look at the top, that's the pathway that I want you to remember. So don't worry about it. If we were to get very specific and technical, yes, from the cochlea, the auditory nerve connects to the cochlear nucleus, and then the superior olive, and then it goes to the inferior colliculus, medial geniculate nucleus, and then finally the auditory cortex. That's a lot of information. So let's try and simplify it and remember these main points. From the auditory nerve, we have remember those bumps we talked about in the midbrain that make up the tectum, the superior and inferior colliculus. Well, the inferior colliculus, or colliculi, if you think about it from both sides, in the midbrain, is a pathway where the auditory nerve, or the information from the auditory system, synapses. So from the auditory nerve, that information goes up to the midbrain to what we call the inferior colliculus. Those are, remember, superior means above and inferior means below. So the inferior colliculus is the um, the little bumps on the bottom side of the midbrain. From the inferior colliculus, remember that 4 of the 5 senses go through the thalamus. So, one thing we didn't cover in chapter 3 is that the thalamus is really made up of a bunch of other little nuclei that are sort of together in the same structure. The medial geniculate nucleus, medial means middle, and genu means bend. So it's a little bent or rounded nucleus towards the midline of the brain, right, where that third ventricle is, kind of near that. And you can see that up top, right above it, above the medial geniculate nucleus. You see that sort of thing that looks like a T. That's our third ventricle where cerebrospinal fluid is flowing through. All right, I don't want to give you more information than you need. So from the auditory nerve through those areas, superior olive is actually in the medulla area. We're not going to worry about that. So for the pathway for you to remember, from the auditory nerve out of the cochlea to the midbrain, which is the inferior colliculi, which helps to convert and cover sound from both ears. As you can see, right before it gets to the inferior colliculus, you see the red and blue pathways sort of crossing over, right? One of the other things that we talk about in the human brain is that all of the 5 senses at some point cross over. So things that are coming from the right ear are going to mostly go 85 to 90% to the left part of the brain and the left auditory cortex. Majority of sound from the left ear is actually going to be interpreted in the right auditory cortex. So that crossover, and again, it's not 100% the crossover, but about 85 to 90% of the axons are going to go in the opposite ear. So most of the sound that comes into your right ear is actually being interpreted in the left brain, and vice versa. Most of the sound from the left ear is going to be in the right brain. So they cross over right before the midbrain, and then from the midbrain, the inferior colliculus, they go to the medial geniculate nucleus, which is the thalamus. Remember that the thalamus takes them to the right spot in the brain to be interpreted, and it also amplifies the signal even more. So you can see we have amplification of the physical signal in the eardrum, in the bones of the middle ear, and in the canal of the inner ear. When they are finally converted, the hair cells bend, and then we get amplification of the neural signal when it reaches the thalamus. In this case, the nucleus in the thalamus is called the MGN, or the medial geniculate nucleus. When we talk about the visual system, we will talk about the lateral geniculate nucleus, or the LGN. And I will try and give you a nice little way of trying to remember which one goes through which. So finally, from the thalamus, it gets projected to where it needs to go, which is the auditory cortex in the temporal lobe. So here we see the pathway from the inner ear to the midbrain after most of them cross over, from the midbrain or inferior colliculus to the medial geniculate nucleus, or MGN, and then finally to the auditory cortex. Now, we talked about the auditory cortex as one sort of place where sound comes in. But if you look at B, you'll notice that it actually gets a little more complex as we are converging into the auditory cortex. The primary auditory cortex is where sound is first entering the brain to be interpreted. But there are different aspects of sound, some more complex than others, and we have to put everything together, like I talked about in the big picture. So there is more than one cortex or area to interpret sound. Sound first gets interpreted, the most elementary parts of sound and the picture being put together, in the primary auditory cortex, or sometimes it's called A1. From there, it goes to a secondary cortex, which may put together different sounds together, different pitches together, so that we can interpret all the sounds in a band or in a choir or in the real world when we hear the wind and horns honking and people talking and blah, 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 maybe now from our own homes. So it gets more complex as we go. And one of the things that allows a very complex system to still be interpreted pretty well is the fact we talked about in chapter 3 that the neurons are arranged in really nice columns and rows, and that makes, millions and billions of neurons at the same time talking to each other. um, it's organized very well, so they're able to do their job, and we can process a lot of things at once. Well, we also see what we call topographical organization. In the auditory system itself, we call that a tonotopic map. So where the sound hits in the cochlea, it is going to hit in an adjacent spot in the auditory cortex. That is what allows that information to come through pretty easily. So let's just say it hits on the inner part of the cochlea. It's going to hit on that inner part in the auditory cortex, and that way you don't lose signal. So we'll get to that in a little bit. Once it goes into the auditory cortex, all of that information, as we talked about in chapter 3, has to go somewhere from the auditory cortex to get integrated in the prefrontal cortex. It's the prefrontal cortex of the frontal lobe that's really putting together all 5 senses. And not only does our auditory system have to interpret what the sound is, but it also has to interpret where the sound is coming from. So we'll talk about how we have specialized cells in our brain that help us to understand where the sound is coming from based on where they hit the ear first. So when we are detecting sounds, that gives us 2 different things we have to look for. One, we have to be able to recognize what the sound is. Oh, that's my wife's voice. She's talking to me. Oh, now she's screaming at me. I need to make sure to answer her or I'm in trouble. Just kidding. So that would be the what. Knowing what the sound is a little bit different than knowing where the sound is coming from. But of course, ultimately, the brain puts them both together. It's just they travel a sort of different pathway in the brain. So for what figure 9.8 shows us is the 2 streams, the where stream and the what stream, known as the dorsal stream and the ventral stream. Now, if you remember from chapter 3, dorsal means up top, right? Towards the top, kind of thinking like a dorsal fin of a shark. Ventral means towards the belly, so that's the bottom side of the brain. So as you see in green, there is a pathway that is traveled kind of on the bottom side of the brain, going from the auditory cortex, which is that big splotch of green on the left, right in front of you, and headed towards the frontal lobe. So it kind of stays low as it goes from the temporal lobe across to the frontal lobe and then up. The ventral stream is telling us what the sound is. So if you hear a sound and you recognize it as a clarinet, you know what the sound is, and that is traveling in that green pathway. For the red pathway, that's where it's coming from, which goes up to the parietal lobe from the temporal lobe, as you can see. And knowing where a sound is coming from, being able to turn your head towards it, being able to orient yourself towards where the sound is coming from, and knowing, oh, that's to my right, oh, that's to my left, oh, that's behind me. Think about when an ambulance is coming down the street and you need to know where it's coming if it's right behind you, because you need to know to pull over or not to pull over, or do I ignore it? Is it something that is going to cause me to make a move, right? Things like that. That's the where stream. That goes up to the parietal lobes. Remember, the parietal lobe is your level of attention, and also knowing where something is in space. So knowing how far or close or in what direction the sound is coming from. So in green, the ventral stream is the what stream, staying low on the ventral side of the brain, knowing what the sound is. Oh, that's a siren. And the where stream is the dorsal stream that goes up through the parietal lobe and then over to the frontal lobe, eventually to the prefrontal cortex. That information, although it travels a different pathway, gets to the prefrontal cortex about the same time, and excuse me, and it interprets both where and what the sounds are. Oh, that's a siren. Oh, it's right behind me. Oh, I better pull over and let it go through. So the what stream or the ventral stream and the where stream or the dorsal stream. You will see the same pattern of firing for the visual system. And a lot of times, of course, we see that the visual and auditory system are very simpatico. They're very, they work well together. They're very compatible. They have very similar pathways, as you will see, and very similar function. And we often see and hear together, right? Watching a movie or a TV show. All of that is putting that information from the visual and auditory systems together. So it makes sense that they have very similar waveforms, very similar physical stimulus, very similar pathways. All right, now let's talk about how we hear the different pitches low and high. In both the auditory system and the visual system, we have a couple of theories that correspond to how we hear the high and low sounds and how our brain interprets them. Both of these theories by themselves are kind of incomplete, and I'll try and explain that. And that's why when we talk about the two different theories, the best way to account for both of them, as you'll see, is to basically a combination of both of them. So here we see that in 1886, there was a man named Rutherford who came up with this idea of the frequency theory. And he said that is the frequency of the waves that tell us how high or low the pitch is, which makes sense. We talked about frequency. The higher or faster the waves, the more waves per second, the higher the pitch is that we can detect. The lower the frequency, the lower the pitch is. So that seemed like a pretty good one. From there, we didn't have a lot of technology in that time, so they just thought about single neurons giving that information. By 1930, that gave way to Weaver and Bray saying that it's not just a one neuron that's giving the frequency, it's actually picking up the frequency from different neurons firing for the different patterns. So that's what we call telephone theory. I don't want to confuse you too much. We're only going to really concentrate on frequency and what we call volley theory. Well, actually, we're really just going to concentrate on frequency theory. So volley theory just says that as this theory was being revised and revised as technology became better, what we found was that more neurons fire in a certain pattern, and so it's the sum of those neurons really that allows the brain to detect the pattern. But one of the things that was realized with volley theory is that sounds above 5200 hertz, and now remember that our brain can interpret from 15 hertz to up about 20,000 hertz. So with frequency theory, this idea of volleying or going back and forth between these neurons was only good for the really low pitches up to about 5,000 hertz. Or actually, I'm sorry, limited to below 500 hertz. So why is that? Why is frequency theory only good for the really low pitches and really doesn't help us to hear the higher pitches? Well, think of it like if you've ever seen RPM on a car or bike or something like that, maybe a stationary bike, and you're looking at RPM, which is rotations per minute. How many times the wheel goes around in a given time, which is kind of like how many waves in a second, right? So if you've ever looked at RPM, maybe on your own bike or a science museum or something like that, you may notice that anything over like 500 or 1,000 hertz all looks the same. We can't individually see all of these rotations per minute. And anything over like 500 rotations per minute all looks the same. 1,000 looks pretty much the same as 20,000. So when our brain is trying to interpret it, they came up with this idea that only the lower pitches are interpreted by frequency theory. So what that means is, what helps us to account for the higher and even the medium types of pitches. If frequency theory is only really equipped for the lower frequencies, there has to be something else going on. So, again, why are these theories inadequate? One is everything kind of looks the same over 500 hertz. And if you remember back to chapter 2, we can't use frequency theory for high frequency because 1000 hertz is the same as 1000 waves per second. Remember that in our action potentials, although they can be faster or slower, we can have more or less in a given amount of time, we still have that thing we call the refractory period. And the refractory period means that at least for 1 or 2 or 5 milliseconds, we can't have an action potential, which means that we can't go over what we already have. Because there's time in between action potentials, this 1 millisecond is well below the absolute refractory period of one neuron. So even though we're getting information from multiple neurons, because of that, and again, you don't have to understand the complete inner workings of this, but because of that, we cannot detect sounds just by using frequency. Is what the theory says. Is we can detect the lower frequencies below 500 hertz, but anything above that, this theory falls short. So, what other theories came into place? Come on. And now my computer is refusing to move forward. It's really weird. Oh, there we go. So let's turn to a different theory that came out. One of the things I told you a few minutes ago and showed you in the slides is that the brain and the ear connections form what I called a tonotopic map. So a tonotopic map says that where the information hits in the cochlea is where it will hit in an adjacent spot in the auditory cortex. So because of this tonotopic map, we know that where it hits in the cochlea is where it's going to correspond to in the auditory cortex. And this gave way to what we call place theory. If you think of the place on the cochlea corresponds to the place in the auditory cortex, then we can sort of map out the level of hertz. In this case, if we look at the base of the cochlea at the bottom, we see 20,000 hertz, and below that 10,000, and a little bit down the line 5,000, 4,000, 3,000, blah blah blah. In the very middle, at the if we were to unroll the cochlea, what we would call the apex or top is actually where the lowest sounds are. So you can see at the bottom in the middle 400 hertz, and at the top of that 600 hertz as it goes up towards 20,000 at the base. So what place theory says is that the frequencies are encoded not by the number of action potentials, but by where they hit in the cochlea. The closer to the base, the higher the sound, higher to 20,000. The closer to the apex or top, the lower the sound. Now, place theory is actually really good for all of the higher or medium frequencies, pitches, because it's where it hits. But where it's not good for place theory is the lower sounds. Why? Because look, at the apex where it's rolled up, the difference between 400 and 600 is pretty big. And if you've ever played a piano, you may notice if you've ever played like the bottom 3 or 4 notes that they all kind of sound the same, right? So it's all kind of muddled at the very bottom. But it's really easy to hear all the sounds in the middle and all the sounds on the top based on which key is struck or which area. And that is very similar to this idea of place theory. It's really good for telling us the medium and high pitches, but it all kind of sounds the same and kind of muddled in that apex area in the middle that's all rolled up. So place theory by itself isn't going to work either, because it really muddles and it's hard to tell the low sounds, but you can definitely interpret the medium and high sounds. So what is the bottom line here? The bottom line is the best way to account for the fact that our brain or our ears, right, to the brain interpret both low and high sounds is, of course, a combination of both frequency and place theory. So again, the auditory cortex is tonotopically organized. So where it hits in the cochlea, as you see on the last slide, anywhere from 400 to 20,000 hertz is going to be tuned to the same adjacent frequency, and it's going to hit in an adjacent spot from 500 to 20,000 hertz in the auditory cortex. And this is how place theory sort of generated all the buzz. But if we are looking on frequency versus place theory, in frequency theory, the hair cells are firing at the same frequency as the sound wave. Volley and telephone just means that you're combining different neurons at the same time to get different frequencies at the same tune. And it works really well for detecting lower frequencies, but not so great for higher frequencies. For pitch and loudness, remember pitch is the frequency, and so that's the impulse rate of the hair cells, and loudness would be the number of hair cells firing. That's how intense it is, right? The more hair cells that fire, the louder it'll be. For place theory, it's where on the basilar membrane the sound hits, because the membrane is sensitive to the different frequencies. Higher frequencies are going to hit towards the base, lower frequencies towards the apex of the basilar membrane. And this creates that tonotopic frequency that where it hits in the cochlea is where it's going to hit in an adjacent spot, as I said, in the auditory cortex. So these are better for detecting higher frequencies and even the medium frequencies. So in this case, the pitch is not the impulse rate, but where it hits in the basilar membrane. And loudness is, of course, the firing rate of the hair cells. So that's the same idea between both of those. So looking ahead, frequency theory alone is inadequate. Sorry, this seems to have combined some information on the slides. So frequency alone is inadequate. Place theory alone is inadequate. So the best way of thinking of it, of course, is, the best way of thinking of it is a combination of frequency and place theory. As you see at the bottom for kilohertz, frequency is really good for the lower sounds. Place theory is really good for the medium and high pitches. So together, the combination of place and frequency theory explains our ability to hear all of the pitches in the human range. Now, we also came across, and I apologize that the picture isn't showing up here, but we also came across that slide at the very beginning that showed pure tones versus what we probably hear in the real world as very complex tones. And we can combine this information with the thing that I said at the very beginning is these sounds come into our ears as individual sound waves, these individual stimulus energies, that bottom-up processing, which means that even though the sound is complete in the real world, when we are interpreting that sound, we have to break it up into its individual little puzzle pieces. Remember sensation, the bottom-up processing? So scientists were trying to figure out exactly how it is that we do this. And we have, as we talked about, different cells that are tuned to different frequencies that combine together to give us the sounds that we hear. So what the brain, a man named Fourier realized is that the brain undergoes this thing called a Fourier analysis. So we have to, in order to interpret, we have to take sounds in the environment and our cells break them up into the different frequencies that fire, and then that information goes back to the brain and basically puts it together again. So we take these complex sounds and break them up into individual components or component frequencies. So the sound is broken up into individual puzzle pieces, sent to the brain, converted into a language the brain can understand, and when they get to the auditory cortex, for that top-down processing of perception, understanding what it is that what the sounds are that are coming into our brain, we put the together into the big picture, put the puzzle pieces back together, and analyze the sounds. So this idea that we break them up into individual pieces of energy and individual frequencies is called a Fourier analysis. So basically what the um, little graph was showing is different frequencies for the clarinet itself. And if you're listening to the clarinet within an entire band or orchestra, that we can hear the overtones. So this way we can hear the entire orchestra or band together as one unit. But if we really concentrated, we could also simply pick out specifically the clarinet that's playing, or the drum sounds that are playing, or maybe guitar or flute or whatever it is. We can hear it all together, but we can also concentrate our attention and focus in on the individual clarinet within the entire orchestra. So, again, if my computer will let me, this is what also allows us to hear not just different sounds within one orchestra, but remember that many different sounds we can be interpreting at the same time. So there might be, when we walk into a restaurant, if you remember doing that, um, or some place where there's a lot of different conversations going on. If you walk into a house party, a lot of times there is the crunching of chips or food. There is people having their own thousands of different little conversations between people. There's loud music a lot of time playing in the background for the party. And not only is it just a mixture of sounds, but eventually, within about a minute or so, we get used to it. So it doesn't sound like a lot of different things, and we're able to hear things. And in fact, um, you may have noticed this is when you get to a party, maybe you don't see your friends at first and everything is going on. And even though there's 1,000 sounds going on at once inside this house, somebody on the other side of the room sees you and calls you, "Hey, Simon. Hey, Bryant. Hey, Tiffany," right? And all of a sudden, even though it's a really, really crowded, really, really loud room, kind of hard to concentrate on one thing, when you hear somebody calling your name, um, we are tuned to the frequency of hearing our own name. So a lot of times we'll turn to wherever and orient our attention to where that sound came from and answer them. Even if your name is called kind of quietly in a very loud room, a lot of times we are able to pick out our own name in a crowded party. And this is what's known as the cocktail party effect. Um, Within that time, whether you're concentrating on music or conversations or somebody calls your name, you are able to turn your attention to follow them, right? So that is um, something we call selective attention. We are tuned, our cells are tuned in our brain to be able to recognize our name, which of course serves an evolutionary purpose if somebody needs to get a hold of you. So not only does this selective attention require remember attention is in the parietal lobe, but it requires directional information too, right? So it's part of our what pathway, and it's also part of our where pathway, right? So you can see the inferior frontal lobe, um, inferior on the bottom of the frontal lobe, is part of that network of selective attention, as well as the posterior middle temporal gyrus. Remember that those bumps on the cortex are gyri. So the middle one, there are three layers. It looks like a superior, a middle, and an inferior area. And it's the middle part of the gyrus in the temporal lobe that seems to be responsible for helping us for this cocktail party effect. One of the last things we'll talk about here in the auditory system is the fact that two ears are better than one. In this case, why we have two ears, so that we can get things from the left side and the right side. And of course, they go to the opposite, mostly to the opposite sides of the brain. So anything coming in the right ear is going to the left auditory cortex and vice versa. And in this way, our ears have to be in sync with one another, because when something comes from your left side and hits your left ear first, it's not automatically it's not going through to the right ear. It actually has to go around or over your head to get to the next ear. So things that are coming from specifically right to the left of you, are going to hit your left ear first, and then they're going to have to go around and hit your right ear. So not only is there a time lag, there may be a difference. So not only is there a difference in time of arrival, but there's a difference in the intensity of the wave. And you might even be in a different phase of the wave, right? So we have the bottom of the wave to the top of the wave. And if it's going to take more time to hit the other ear, we're also looking at a difference in how loud it is. So a difference in intensity and a difference in the phase, what part of the wave that it's in. All of these things would make it really annoying if our brain didn't have a way to compensate for that. You would essentially be hearing an echo in your own head, right? It'd hit the left ear first and then the right ear about a half a second later or something like that. And you'd hear everything kind of with an echo. And that would be, if our brain couldn't adapt to that, one of the worst ways to get through life. Although if it was always like that, I guess maybe by now you'd be used to it, right? So what does that mean? Well, again, you can't see the picture, and I apologize for that. Higher frequencies create what's called a sound shadow um, made by our head. And so what we get is a difference in intensity. In the higher pitches, it hits one ear louder than the other. At lower frequencies, it's because, again, it takes longer for lower frequencies. We have less wavelength, less hertz. So we get a sound delay because it's a difference in the time of arrival. So in both higher and lower frequencies, if we did not have a way to correct this, it would be really, really hard to go through the world hearing things in echoes. But fortunately, and here's what I mean by the phase difference. So the far ear's wave will lag behind the nearer one. If you look at the difference for the woman standing there, it's only useful below 1500 hertz. So lower sounds, if you look on the left, it's sort of in the, for her right ear, it's sort of in the middle of the wave by the time it hits it. And for the left ear, it's definitely down at the bottom of the wave. And so you'd be getting different sounds in each ear. At higher frequencies, it seems to be okay. It's about the same phase. It's not as big a worry, but what this shows us is that the brain has to have a way to take care of that. We don't want to have an echo. We don't want to have a different, tone or intensity, different wavelength phase from each ear. So there has to be some sort of system inside the brain that compensates for this sound shadow or sound delay or for these lags in phase difference or intensity. And the answer, there we go. Is that, nope. The answer is called coincidence detectors. So I'm going to come back to this, and I always meant to move this in my slideshow, and I never did, but let's get back to this one. So coincidence detectors, and again, I apologize, I'm going to send you a picture of these coincidence detectors along with the other pictures we're missing for this format of the slides. I don't know why it does this. I guess it's a conversion problem from the original slideshow. But these coincidence detectors are basically specialized cells in the ear, in the brain, that take the sound that goes from one ear to the other and compensates for the distance and the lag and the differences in phase and intensity. So what you would see and what you will see when you look at the picture itself after you listen to this probably is that you have A on one side. So when it hits the left ear first, it's going to take a longer time, and we're going to cut that with the right ear. If it hits from a different angle, it gets compensated basically. So you'll see it in the picture. I'm sorry, it's hard for me to explain it without seeing the picture in front of me as well. When somebody is talking right in front of you or right behind you, it's going to hit both ears at the same time, so you won't need to worry about any lags or anything like that. That's how we know when someone's talking right in front of us, is it hits both ears at the same time, and then you don't have anything to compensate for. If it's at an angle and hits the left ear first, it's compensated on the right ear, So basically, these coincidence detectors allow for us to hear things without that annoying echo and interpret them. But because it hits one ear first and that information is sent to the brain knows in that dorsal stream or the where pathway, it knows where the sound is coming from. So if you're at that party and somebody calls your name, not only can you hear your name over all of the other noises, but you can also orient your attention and look at the location of where that sound came from. And that is the coincidence detectors. So really quickly going back, one of the things that we talk about when we convert a physical stimulus like a sound wave into a neural signal that the brain can interpret is that's how that information gets there. So to say that somebody that doesn't have um, or has problems with this system is deaf, most people think, without knowing the pathway from the ear to the brain, that the cause of deafness has to be in the ear, right? And it's true that that could be the case. But with sensation and perception, nothing is actually perceived until it actually reaches the brain so that the brain can put it together in the big picture. If you have perfectly working ears, but somewhere along that pathway from the ear to the brain gets interrupted, if that auditory nerve gets cut, or if the signal can't make it to the brain because the auditory cortex is damaged or things like that, you're still never going to hear anything, even if your ears are perfectly functional. Um, and a lot of times, the deafness comes from the inability of the cochlea. Maybe it's the hair cells that are damaged or something in the inner ear, but it's the inability of the cochlea and the hair cells to convert the physical signal into something the brain can interpret. And if that's the case, so that's anywhere from the hair cells to um, the actual auditory cortex, if there's a problem there, it might be just a problem of not being able to transduce or interpret the signal. So sometimes you may have seen kids or adults that have this thing that looks like a transmitter, um, an antenna above their ear, and that is called a cochlear implant. And what a cochlear implant does is, because the signal is right that the hair cells give off is an electrical action potential, what the cochlear implant can do is it gets threaded into the cochlea and it can basically stimulate the hair cells directly, um, or interpret what the vibrations are sending to the hair cells and converts the signal for the hair cells if they can't do it. And in this way, the brain still gets the message. So it is a way to repair that deafness that comes from what we call nerve deafness, that is affecting either the hair cells or the corresponding auditory nerve. And so that signal which was interrupted can now make its way to the auditory cortex, and effectively the person can hear. It's an amazing invention. As you'll see, they also have something like that for the retina in the eyes. I don't know if it works as well, but this is pretty cool. And I will tell you that the first time I taught this class at ELAC, somebody showed me a picture. This is when my son was about 2 or 3, and I think he was 3 at the time. And it was a little girl who was about 4 or 5 years old that had to have, for this reason, had to have a cochlear implant. And her father did not want her to get made fun of. So he shaved his head and he had a cochlear implant tattoo um, put on the same side of his head as his daughter had, so that she would never feel alone. He would always have that with her. And of course, at the time, I was like, no, I'm not going to cry. Well, uh, let's just say I won't finish that story, and you can probably figure out why. So it was very sweet, and it always reminds me of that. So that is the end of the hearing portion of the chapter. So I will give you guys a little break for my voice, and I'll catch up with you on the next portion of this chapter, which is language. I will talk to you very soon.